कि माइक्रो ऑर्गेनिजम्स क्या होते हैं। माइक्रो ऑर्गेनिजम्स बेसिकली वो ऑर्गेनिजम्स होते हैं जो बहुत स्मॉल होते हैं और जिन्हें हम अपनी नेकेड आईज से नहीं देख सकते। उन्हें देखने के लिए हमें माइक्रोस्कोप की जरूरत होती है। ऐसे स्मॉल माइक्रो ऑर्गेनिजम्स को हम माइक्रोब्स भी कहते हैं। बेसिकली 4 टाइप्स के ज्यादा हार्मफुल माइक्रो ऑर्गेनिजम्स होते हैं। फॉर एग्जाम्पल बैक्टीरिया, वायरस, फंगस और पैरासाइट्स। ऐसे ही और वीडियोस और शॉर्ट्स देखने के लिए लाइक करें, कमेंट करें, शेयर करें हमारा चैनल।
Hello student, today we discuss about the introduction to food safety and the microorganism that spoil the food or comes under the food microbiology. So today we discuss about the introduction to food safety and microbiology. So first of all, what are the food safety or food hazard? Actually, it is divided into a causative categories called a hazard. The organism microbes which spoil the food, right? Which spoil the food, or some microbes which are helpful in the food industry, helpful for the food industry, that also comes under the food microbiology. And they are the microbes which cause or spoil the food, that comes under a hazard. So there are two types of microbes. First microbes, there are two types of microbes. First one which help in the food production, right? And second one which spoil the food, right? Spoil the food, or that comes under a hazard. These all are the two types of microbes. Clear? So biological hazards are bacteria, molds, naturally occurring toxin. These are biological hazard. And chemical hazard are chemical like petroleum, herbicide, pesticide, heavy metals. These are the chemical hazard. And physical hazards are glass, rocks, wood, splinters. This comes under physical hazards. So in this video we mainly discuss about the biological hazards that happen by the microbes. In 1919 versus 1997, the hazards caused by the microbes are tuberculosis, pneumonia, diarrhea, heart disease, liver disease, injuries, stroke, cancer, bronchitis, diphtheria. So the ratio of the disease in 1919 to 1997 is more as compared to 1999. In 1997 it is more. In the 10 leading causes of death in the United States in 1900 and 1997. Clear? So microorganisms such as bacteria, parasites, virus, yeast and mold, these all are the microorganisms that seen via a naked eye. Clear? This is the chart of microbes. These are the atom molecules: virus, bacteria and archaeal organisms, unicellular algae, colonial algae, protozoa, fungi, multicellular organisms. These are the microbes. Actually, where these microbes are found, they are present everywhere in our environment. Everywhere. We can't say that here there is no microbes. In our environment, everywhere there is a microbe. It's either soil and water, it is utensil, equipment, gastrointestinal tract, food handlers. In everywhere having a microbes. Sources of fecal contamination of foods. This is the main sources of the fecal contamination of foods, because there are a house fly, right? House fly, mosquitoes, these insects or flies, what they do, they sit everywhere. It is that fecal is present there or food is present there, they sit everywhere, and contamination occur by these insects. So what happened? Meat animal, processing plants, product contamination, food ingredient. What happened? From poultry, dairy cows, humans, sewage, everywhere, from everywhere where this food contamination, product contamination will be occur. Microorganisms in food. Microorganisms are important in many different ways. They are important, already I told you, microorganisms are important and harmful. Both are present in a food industry. So how they are important? How they are important? Pathogenic or disease causing microorganisms can cause illness. These are the important. No, this is not important. This is the actually for microbes they are important. In terms of microbes, this pathogenic, their activity are pathogenic. So for microbes it is important. They spoilage microorganisms cause of food to smell, taste and look unacceptable. Fermentation microorganisms produce desired food product. So pathogenic and spoilage, these are the harmful effect of the microbes, whereas fermentation is a useful effect of the microbes. Other microbes do nothing in foods. So these are three different ways how they are important and how they are harmful. So pathogenic and spoilage, these are a harmful effect, and this fermentation is the useful effect of the microbes. Clear? Next is controlling growth. How the microbes are controlled. The microbes for food nutrient content for growth, microorganisms require the following. They also require a substance for their growth, for their metabolic activity, because without the metabolic activity there are no organisms who are survived. Either they are microbes or prokaryotes or eukaryotes, they all having a metabolic activities for their division, for their survival. Clear? And for the metabolic activity, they require this material like water, source of energy, source of nitrogen, vitamin and related growth factors and minerals. Understood what they require? They require water, source of energy, source of nitrogen, vitamins and related growth factors, mineral. Now food activity. Optimal pH for the growth of microbe is 6 to 8. What? The optimal growth for the growth of microbes is 6 to 8, and for the disease causing bacteria it is 4.6 to 9.5, and for the spoilage bacteria it is 1.5 to 9.5. These are the activity of the microbes according to their pH. Now pH growth range in foods. Moles having survive at the range of 11. Yeast is from 1.5 to 9.5. Lactic acid bacteria, they are active at a 3 to 12. Staphylococcus is from 6 to 10.5. E. coli is in acidic medium and basic medium, both they can be survive. But Vibrio species survive at a neutral, approximately survive at the neutral pH. The mild heat required since spores are inhibited by acid. High heat required in order to destroy spores. Controlling growth. With the help of temperature, we can control the growth of the microbes. With the help of nutrient or food, we can control the growth of the microbes. And according to pH, also we can control the growth of microbes in the food industry. For the optimal growth of the microbes, the temperature is 37 degrees Celsius. But there are some thermophilous bacteria are also produced. Microbes are also present that grow in hot condition. Mesophils like warm condition around body temperature. Psychrotrophs can grow at refrigeration temperature, and most pathogen are mesophils. Temperature classification based on optimum temperature for growth. Optimum temperature for growth: psychro cold, meso means middle, thermo means warm, tropic means growing, diuric means withstand, phil or philic referred to love, means they want to grow at low temperature, they want to grow at high temperature. Clear? And these are the graph at which the bacteria will grow. This is the temperature, and this is the hyperthermophil which grow at high temperature. This is thermophil which grow at high temperature but not that too much high. And mesophil grow at normal temperature, and psychrophil at a low temperature. Clear? Controlling growth: temperature lower than 41 degrees Celsius. Temperature that is lower than 41 degrees Celsius, we can control the growth. Bacteria ceases to multiply but do not die. Freezing can cause cell wall damage. To damage. Temperature higher than 135 degrees Fahrenheit, the bacteria will be die if heated for a sufficient time. And if the temperature will be increased at the 135 degree Fahrenheit, the increased destruction with longer times and with high temperature. Now time. Under optimal conditions, some bacteria can double every 20 minutes. The colder the storage temperature, the longer the potential shelf life. Potentially hazardous food should not remain in the danger zone for more than 4 hours during the entire food handling process. These are the diagram of how the bacteria will be divided in every 20 minutes at the formula of 2 to the power n, because every time they double from the initial step. Clear? Now generation time under optimal condition. At 37 degrees Celsius, Bacillus species, the generation time of Bacillus species is 28 minutes. And for E. coli, the generation time will be 12.5. And for Staphylococcus species, it is 27 to 30. Clear? And this is the growth phase of the bacteria. This is lag phase, this is log phase, and this is stationary, and this is the death phase. How the bacteria will be growth and how they will be die. In lag phase, they require nutrient, they start dividing but not too much. In log phase, they divide and increase their number. In a stationary phase, they form a static condition, and in death phase there is depletion of the nutrients, so they are die. Oxygen is one of the another important factor for the microbes for the growth, and how we can control the growth of microbes with the help of oxygen also, because aerobic bacteria need oxygen. If we don't supply the oxygen, then Aerobic bacteria will not grow at that in that environment. Anaerobic bacteria go only if oxygen is absent, and facultative will be at both. So tolerance to oxygen in the surrounding environment, according to tolerance, there are 3 types of bacteria. Next is water activity. Moisture in a food system that is available for microbial growth and chemical reaction, the relative humidity of a food. Clear? This all are a controlling growth active, water activity of common food. What? Water activity of common food. Now, what are the controlling factor from which we can control the activity of the microbes? That is a FATTOM. That is, F is food, A, right? F is food, A is acidity, T is temperature, and next T is time, O is oxygen, and M is moisture. So these are the term by which we can control the growth of the microbes in food industry. Clear? Now, intervention, combined effect. Predictive microbiology, the study of interactive effect of factor affecting microbial growth. Single factor is also affect the microbial growth, and there is a if we want the effect of 2 or more factor at a time, or the interactive effect, then this effect is more effective as compared to single. So additive effect, there is synergetic effect and antagonistic effect. There are 3 types of effect in food industry. Food industry interventions. Ingredients have good specification and control of incoming ingredients. What? Ingredient having good specification and there is control of incoming ingredient. They having the process of an adequate thermal process to destroy microorganisms so that they not spoil the food. Have an adequate packaging system to protect your product. After processing, when processing will be completed, have control of distribution, and a system for tracking and recalling. Provide necessary information for consumers. What they provide? They provide necessary information for consumer. And farm to farm food safety and quality program. Ingredient control. How we control the ingredient? Specification for ingredient makeup. We consider the quality, physical and microbial contamination level of the ingredient, how the ingredient are affected, which microbes affect which ingredient, we have to know. And during the processing, we have to control the effect of the temperature, air, according to that, and control the activity of the microbes. So, a specification of ingredient makeup, quality, physical and microbial contamination level. Clear? Thermal processing control. In thermal processing control, we control the cooking, pasteurization, commercial sterility process, retort system, aseptic processing and hot filling. Package integrity. In package integrity, measurements are made to ensure the package is of food sanitary quality and can maintain a hermetic seal, such as visual inspections and torque measurement for your container. During the packaging, there is much necessary to see the hermetic seal. Clear? To see the hermetic seal. And during the thermal process, there is necessary the pasteurization, commercial sterility. Clear? Thermal processing control. In thermal processing control, low acid food should have a process to eliminate millions spores of Clostridium botulinum. Example, most meat, vegetables and dairy product. Process often at 230 degree Fahrenheit or above. Means there is a high temperature. It is a thermal processing control. How we control the microbes here? With the help of thermal. The bacteria which grow at low acid food. And in acidified food, not require too much a process to eliminate Clostridium botulinum. Example, pickled products, mayonnaise, for acidified food, pickled product mayonnaise are required. Process often 180 to 205 degree Fahrenheit. Pasteurization, hot filling. Last one is high acid food. For high acid food, they not required to have a heat process. Example, sliced orange, condiments. Process often all occur at a high temperature, so that bacteria will spoil at this temperature. For low acid food, the temperature that are required is 230 degree Fahrenheit or above. For acidified food, the temperature will be at 180 to 200, and for high acid food it is 180 to 205 degree Fahrenheit. Now pathogen. General role of microbes that present in a food microbiology. There is a commensal, ubiquitous, harmless or beneficial. Already I told you the microbes are of two types. They are harmful or beneficial. In spoilage, the food in spoilage what happened? They cause or harm the food. The bacteria, microbes which cause the harm of food. So cause food to become inedible due to change in color, flavor, odor, appearance or texture. Grow to high levels and break down food component. What happened here? In spoilage, the bacteria grow to high levels and they break down food components for their own utilization. Commensal organisms that have reached a high population. Different products have different spoilage. Different products have different spoilage flora. Not single organism can only cause a spoilage of the food. There are different types of microbes. They cause a spoilage of food according to the type of food or according to the temperature that are present in environment. So the beneficial effect of the microbes are used as an aid in producing desirable characteristics in food, such as fermentation. Fermentation is a useful term or useful food that are break down or produced by the microbes. Like for example, bread, idli, dosa that are fermented with the help of the yeast, right? So these are a useful product. Vinegar. Pathogens cause foodborne illness. These are a harmful effect. Pathogen cause foodborne illness. Microorganisms that are usually associated with the presence of pathogens are called indicators. Most E. coli do not cause illness. Food safety control hazardous to the consumer. What food safety cause hazardous to the consumer? A foodborne hazard is biological chemical or physical properties that may cause a food to be unsafe for human consumption. Food quality control deterioration of food to attain an unacceptable state. Food safety control hazards to the consumer. A foodborne hazard is a biological, chemical, or physical property that may cause a food to be unsafe for human consumption. Food quality control deterioration of food to an unacceptable state. Foodborne illness. Foodborne illness is in the United States is associated with 46 million illness in a year and 3,25,000 hospitalization in a year, 3,000 death in a year. This all data is from the foodborne illness. Common foodborne pathogen. In bacteria, the pathogen are E. coli, Salmonella species, Staphylococcus, Shigella. These are bacteria. And virus, norovirus, rotavirus, hepatitis C. Parasites, Giardia, Cyclospora, Cryptosporidium. 80 to 90% of foodborne illness from bacteria come from just 4 bacteria, that is Campylobacter, Salmonella, Clostridium perfringens, and Staphylococcus aureus. These are the main 80 to 90% of the foodborne illness factor that responsible for the foodborne disease. Illness mechanism. First, infection. Microorganisms are ingested and then cause illness. Intoxication. Toxins are produced by the pathogen usually in the food. When food is consumed, illness occurs. Even if microorganisms are killed, toxin can still remain the food. So that the food eaten by the people, the toxin go inside the body and they cause a illness, and that illness is known as foodborne illness. Bacteria, already I told, the bacteria that are cause foodborne illness are E. coli, Salmonella, Listeria, Campylobacter. Clear? And these are the bacteria, how much cases and death? E. coli का as a hemorrhagic colitis, cause infection, incubation period 2 to 4 days, symptom diarrhea, HUS, TPP, and contaminants are milk, meat, fruits, vegetable, water. Clear? Salmonella cause 10 foodborne illness, incubation period 6 to 48 hours. What? 6 to 48 hours. Symptom, nausea, fever, diarrhea, arthritis, contamination, milk, meat. Next is Listeria monocytogenes. Same here also. The symptom is different. Here vomiting is also comes in the symptom. Vomiting, diarrhea, meningitis, septicemia, miscarriage. Contaminants, vegetables, milk, cheese, meat, seafood. From this all we see that meat and milk are most contaminant food material that contaminated by the microbes. Clear? So when we eat these two items, we clear that we heat them before we eat or drink this at high temperature, so that the microbes present in the milk and meat will be destroyed. Next is Staphylococcus aureus, cause intoxication, incubation period 1 to 6 hours, symptom nausea, fever, diarrhea. Another is Clostridium botulinum. They cause foodborne disease. Prevention of foodborne illness. Cook the food. Cook all meat, poultry and egg to at least 160 Fahrenheit or high temperature. Other than spore forming bacteria or all bacteria or microbes are killed quite easily with the heating at 160 Fahrenheit. Avoid cross contamination. Do not cross contaminate one food with other. Another, keep raw food totally separated from cooked food. Chilled food, keep foods cool. Keep foods cold after cooking. Chill foods are rapidly as possible. Remember that cooking as destroy most of the bacteria, but spore forms that are resistant to cooking may become very active and can proliferate rapidly. Cleaning, wash fruit and vegetables and all food possible. Personal hygiene. People working with food should wash their hands regularly. Wear hairnet, plastic gloves, etc. Spoilage organism, already I told you, bacteria, fungus, protozoa are a spoilage organism. So this all are about the food microbes that are present in food. Okay, hope this all are clear. So today we stop here.
Today I am taking a class on contamination, preservation and spoilage of the food. Okay. So as you know that microorganisms are present in all around us, and definitely they will give the effect to our food content also. And microorganisms are so we can say that microorganisms are associated in variety of ways with all of the food that we eat. They may influence the quality, ability, quantity of our food. And the major thing is that they are naturally occurring foods such as fruit and vegetable. They are normally contain some microorganisms and may be contaminated with additional organism during the handling of all these things. So the food can be served as a medium for the good for and medium for the growth of microorganism and their growth, and may cause the food to undergo the decomposition and spoilage also. Got it? So food may also carry pathogenic organisms and result that can transmit the disease also, and some other microorganisms that allow to grow in the certain food products, they can produce the toxin substances, and that is result in the food poisoning when the food is ingested. And other microorganisms that are used for the preparation of the different type of food products such as yogurts and curd, so that is also associated with the formation of food. Microorganisms are we are using for the preparation and preservation of the food also, like yogurt and other things. And generally we can say that major concern in this because of we have to discuss about the food microbiology. That means normal flora of the food, their significance, their manner in which the food can be protected, and microbial contamination, microbial spoilage, and the things by which the food can be affected. So the microbial flora of the fresh food we can discuss here. So first we are started with the introduction also. Mainly we will see the different changes in cereal and food products. Important food resources are livestock feed worldwide. The exterior and harvested grains retain some of the natural flora than while growing, plus contamination from the soil and insect and some other sources. So the freshly harvested grain that contains the load of thousands of microorganisms and mold also. So for the preservation, what we do, we will do the removal of the excess moisture in the cereal, grain and flours and other things, and preserve our food as in a dry things. So due to the drying of this process, the microorganism cannot grow on this, so we can avoid the spoilage and contamination of the food. Okay. So main microbial fresh food, first we will study about the main microbial flora of the fresh food. Okay, so inner tissue of the healthy plants and animals, they all are the free of microorganism. And the surface of raw vegetable and meats are contaminated with variety of organisms. You should know about that. And the magnitude of the microbial contamination that reflects more or more of the formation of the microbial population of the environment and from which the food are taken, the condition of the raw product, the method of handling, how we are handling the food, the time, which time we are collecting the food, and the condition on which we are storing our food products. Okay, so it is desirable to maintain the very low microbial level of contamination on the raw food and the presence of extremely large number of the microorganisms, that some undesirable events can occur and that the food is indeed susceptible to further deterioration. Okay, so main food products are the cereals, meat, poultry, eggs, fruit and vegetables, and shellfish and finfish, milk, and some other biochemical type of bacterial milk. Okay, nowadays a lot of things are going on, and how we can characterize the bacterial retention in the milk and different type of reactions, and what kind of organism that can contaminate the milk flora, and how we can preserve all these our food product, and what are the spoilage that we will discuss in further in this chapter. Okay, so first I am starting with meat. So the carcasses of healthy animals, that is of slaughtered by the meat and held in the refrigerated room and likely to have only the nominal surface contamination, and while the inner tissues are sterile. Okay, when the fresh meat cut from the chilled carcass, its surface contaminated with a microorganism and characteristics of environment and the implement, that is saw and the knives that is used to cut the meat. Each new surface of the meat result from the new cut and add the more microorganism to expose the tissue. The ultimate in providing the new surface and potential contamination of the meat surface occur in this process of making the meat as a food. So, to improve the microbial quantity of the meat, particularly ground beef, and most states have to adapted standards and that are considered established the regulation to require the microbiological standards and the things which we had purchased from different type of outlets. Among the most common species of the bacteria that are present on the fresh meat are Pseudomonas, Staphylococcus, Micrococcus, Enterococcus, and coliform. And at the low temperature, where the fresh meat are held, that favors the growth of phycophilic microorganism. So that is about the meat. And second, we can discuss about the poultry. So the poultry, the freshly dressed, the poultry have a bacterial flora of their own surface, that is of skin, and that originate from the bacteria normally present in the live birds and from the manipulation during the killing, defeathering and aviations. And under the good condition or good sanitary conditions, the bacterial count are reported very less bacteria per square centimeter of the skin surface, where where under the less sanitary condition the count may increase 100 times folds or more. So main organisms which are present on the egg and the other poultry things are the Pseudomonas. That is a major contaminant of the skin on the freshly dressed poultry. Okay. Next is egg. So the interior of the freshly laid egg is usually free from microorganisms. Okay, microorganisms effect are vary in the minimal amount, and that is also depends on the sanitary conditions, which is the condition of the storage, what is the temperature, what is the humidity. Microorganism, particularly the bacteria and the molds, that may enter into the cracked egg or in the shell or penetrate the shell when the bloom, that means the thin protein coat that is called the bloom, that is covering the shell deteriorates. Okay, the type of microorganisms that involve the reflect those present in the environment. Next is a fruit and vegetables. So fruit and vegetables are normally susceptible to the infection by bacteria, fungi and viruses. And microbial invasion of the plant tissue can occur during the various stages of the fruit and vegetable development. And that's why the tissue are invaded and spoilage is very much increased. And the second factor that contributing the microorganism contamination of the fruit and the vegetable, that pertains to their post harvest handling. Okay, and mechanical handling is also produced, that is breaks in the tissue also. The pH of the fruit is relatively acid, that is ranging from the 2.2 for the lemon and 5 for the bananas. This restriction bacterial growth but does not retard the fungal growth. The pH that range from for the vegetable is slightly higher, that may be the 5 to 7, and that's why these are more susceptible than fruits to attack by the bacteria. And next type of food is that is called the shellfish and finfish. The microbial flora of the fresh these both fishes, because they are the aquatic, and these are very largely reflection of the microbial quantity of the water where they are harvested, and of particular significance where the water is sewage polluted or sea or that we have harvested from the seafood as a seafood. So main bacteria that can be cause the spoilage and that can be present, that is of Vibrio cholerae and some other species of the Vibrio like Vibrio parahemolyticus, that is the mainly causes the gastroenteritis and epidermic infections in different persons. Okay, and these seafoods sample that include the fish, shellfish and other that grow in the contaminated water can concentrate viruses and may be source of the hepatitis infection, and like oysters, clam to polluted waters, and that causes the numerous epidemics in the various part of the world. Next, that is very much important, is called the milk. So at the time of we drawn the milk by the udder of the healthy animal, the milk contains organisms that have entered the teat canal through the teat opening. Okay, so they are mechanically flushed out by during the milking process, and the number present at the time of milking has been reported to the range between the several hundred to several thousand millimeter. The count vary among the cow to cow and the person who are handling the cow, that is also depends on that. Okay, and from the time of the milk leave, the under the when we dispenses to the container, everything which come into contact with the potential source of the microorganism, and that includes the air contamination that is comes from the environment, and milking equipment that we are using for the milking process, and some other the handlers personal hygiene and sanitary condition, that also depends, and um, that also causes the milk spoils rapidly. Okay, so milking is generally performed under the hygienic condition, very strict attention to the sanitary practices, and that will result in the product of low bacterial content and good keeping qualities. Okay, next we can say about that the basis of their major characteristics, that is called the biochemical types we can check, temperature resources we can check, and ability to cause the infection and the disease that we can do. Okay, next we comes on the cereals, that bacteria that in the wheat flour that include the bacillus, coliform bacteria, some other bacteria like Sarcina, Micrococcus, Flavobacterium, Serratia, that all causes the contamination in the cereals also, and sometimes the molds are also involved. The mold spores are chiefly of the Aspergillus and Penicillium, and with Cladosporium and some other genera, and aspergillus flavus and aspergillus aflatoxins that produces the aflatoxins also. Okay, about that, different type of flours have a different type of contamination. The cornmeal flour that contains several hundred to several thousand bacteria and molds per gram, and fusobacterium and penicillin that also causes, and because of the incubation of the moist condition like malt containing the high number of the bacteria, and the surface of freshly baked bread that is a free from all viable microorganism, but when we put it on the some room temperature, that can be contaminated by the mold spore, by the air during the cooling and before wrapping, and the cakes are similar subject to contamination, and spores are when with the appropriate conditions is there, so spores can be germinated to the vegetative cells and they will produce the spoilage of the food. And the contamination of the cereal grains, that is also affected by the different type of molds, because the molds contain the different type of mycotoxin that is responsible for the different type of disease in the human being. So about that, first we have discussed about what kind of organism, what kind of food and what type of contamination they are producing. Secondly, we will discuss about the spoilage of the food. So the considering the variety of natural food substances and method by which we are using, handling method which we are using, so different type of organism and different kind of microorganisms are potentially contaminant, and the type of food substance and the method by which it is processed and the preservation method can be used for the different organism to prevent from the spoilage. Okay, so most foodstuffs serve as a good media for the growth of many different microorganisms, and they will give the chance to grow the organism that will produce the that will produce and change the flavor, odor and qualities of food. So they will degenerate the substance and they will produce some certain type of substances, like in the putrefaction process, the protein food, that proteolytic microorganism that will change into the ammonic acid, ammonia and amines and some hydrogen sulfide and some this sulfur molecules that is responsible for the smell and putrefaction process. Next process that is called the fermentation, that is caused by the microorganism in which the microorganism will affect on the carbohydrate food, and carbohydrate fermentation microorganism that will change into the acid and alcohol and gases. Third type of process that is called the rancidity. The rancidity is a process in which the fatty food and lipolytic microorganisms work on it, and that will change into the fatty acids and glycerol. The changes that microbes that can cause the food that are not limited to the result of degradation. They may also be caused by the product of microbial synthesis. Some microorganism that is discolored food as a result of pigment production, and slimes that may be developed in or on food by the microorganism that is capable to synthesize the certain polysaccharides also. So if we want to discuss about the fresh food, so how they spoil, what are the organisms, so we can discuss here in short. So first organism we can say that is a bread, and the type of spoilage is moldy, and main organism that is involved are the rhizopus and penicillin and that is aspergillus. Okay, and maple sap and syrup that is destroyed by the um, antrobius bacters, Saccharomyces and zygomyces, and the most organism that is responsible is a that is a saprophytic molds and yeast, and type of spoilage is ropy and yeasty. They are pink, they change this into the pink and moldy, and all these things we can prevent by the different methods. About the cereals, we can use the low moisture content, so little difficult to preserve the growth of microorganisms, but we can prolong the deterioration and spoilage. And the mainly the temperature on which we can store the food is on the 4.4 to 7.2 degree. Okay, and some other product like bakery products like bread, rolls, cakes and pastries that have enough moisture, so these foods are easily, and they are good source, they are good providing the good energy source and food to the microbes, so they are the main effective foods for the microorganisms. So we have to give something to prevent this. Okay, next type of food we can include is that pickle and fresh meat. About the pickle mainly the type of spoilage is film or the yeast. Sometimes you are find out that some yeast kind of film is make on the pickles. And on the fresh meats, putrefaction is there, and main organism that is responsible is a Aspergillus niger, Rhodotorula. And about the meat putrefaction, there is an Clostridium, Proteus vulgaris, and some species of the Pseudomonas. Next type of food is that is called the cod meat, that is of spoilage is about the moldy, and main organism that cause the spoilage is Aspergillus, rhizopus and penicillin. And generally they have producing organism has producing the greenish or the slime like substance that is caused by the lactobacilli and some micrococcus also. Okay, next type of organism we can say that fish is of spoilage is like a discoloration, putrefaction, and the organism which is responsible for that is Pseudomonas and some Flavobacterium. Next food is called as axe, that is of green rot, spoilages and colorless rot, and the organism which is responsible is the Pseudomonas and some Proteus that causes a black rots. Okay, and concentrated organic juices, they will change into the off flavor, that is due to the presence and growth of lactobacilli and some acetobacter. And other food like we can say this is the poultry that will change into the slime and odor, so that these organisms like Pseudomonas and some allicogens are responsible for the this type of putrefaction. Okay, so this is about the all fresh food and their contamination and their spoilage. Before studying about the prevention, we should have an idea about the canned food spoilage also. So the different type of product like low and medium acid products, and the product that pH level is about 4.6 like corn, peas, spinach, asparagus. So the type of a spoilage that is of flat sore, okay, that is produced by the Bacillus species, thermophilic anaerobes, Clostridium species, sulfide spoilage, and aerobic spoilage. So cane how we can identify the in the cane possible loss of the vacuum on the storage. Okay, and content of cane is appears not usually altered, pH markedly lower, sour, may have a slightly abnormal odor, and sometimes cloudy liquids may be present. And second type of food is that is called the acid products like pH lowered from 4.6 like tomato, juicy fruits and fruit juices. And the type of spoilage is flat sore that is caused by the Bacillus thermophilus and Butyricus anaerobis, Clostridium butyricum, some yeast and some molds. And we can see in the cane is swell, may be burst, can swell usually burst, and can swell may be burst, and sometimes cane will be changed into the flat. Okay, fermented butyric waters, and what will change acid odor, fermented yeasty order, and surface growth is musty order. So this type of changes that can be present in the different type of food. So the cane food, about the cane food, because of the heat resistant spore formers like Clostridium and Bacillus that constitute the most important group of the microorganism in the canning industries. And the three most important type of microorganism spoilage of the commercially canned food are flat sore spoilage and thermophilic anaerobic, that is called the TS spoilage, and third one is called as putrefaction. Three type of spoilage is there that is produced in the cane food. Okay, and this that all involves the spoilage of cane food together with a change in their products. Okay, so when we examine for the microbiologically is all these cane food, so we will obtain something. And so first we will discuss how we can prevent it. So the main thing is asepsis, that is very much important, that is sanitize the equipment, that is maybe source of many bacteria, and they mainly forming bacteria that causes a sourceness of the bread, cake and other baked products, and that can be storage by the different type of molds, and that is contaminated by the mold spore. How they can prevent? By the use of heat. We can use the heat in the different variety to prevent the all kind of contamination and for the all kind of we can say that spoilage. Okay, so mainly the food preservation generally which we are doing in the refrigerator, deep freezer, and sometimes we are using the temperature also. Nowadays we are also using some lyophilization technique. So the modern method of the food preservation, that is mainly based on the aseptic handling and high temperature, that is of boiling, and high temperature that also include the steam under the pressure, pasteurization, sterilization of the milk and aseptic process. And next is a low temperature by the refrigeration, freezing, and by the dehydration, by the osmotic pressure, by in the osmotic pressure the concentrated sugar and with a brine. And in chemical we can also use the organic acid substances that developing during the process, that is called as smoking, and substances that is contributing by the microbial fermentation, that is acid. And next method we can also use for the preservation is a ultraviolet light and ionizing radiation for the preservation of different type of. So first is a use of a different type of heat. So the high temperature is one of the safest and most reliable method of the food preservation, because heat is widely used to destroy the organism in the food products like in canes, jar and other type of condition that restrict the entrance of microorganism after the processing. So the steam under pressure such as pressure cooker is most effective method for the high temperature food preservation, and that will kill the all vegetative and the spore organism. Okay, and the food preservation by the heat, that is required the knowledge about the what kind of food it is there, what kind of bacteria can be present. So that depends on the knowledge of all these things, and we have to have an idea about that this food is containing spores or not. Okay, in addition one must consider the rate which the heat penetrate through the food of the different consistencies, about their size of container and type of fluid. So the killing of microorganism by the heat, that involves some time temperature relationship and product that we are using for the sterilization. Okay, and generally bakery product may be the solid. So some products are in liquid form. So lot of things are there, but before processing we have to just have an idea about the all kind of things. Next type of method we can also use it as a canning method. The temperature used for the canning food range are almost about the 100 degree centigrade to 125 degree centigrade, and for the low acidic food. and the canning process does not guarantee that style products. Okay, so sometimes spores may be present that can be germinated. Mostly the important organism is a Clostridium botulinum. About the milk, so most important thing that is called the pasteurization. So the pasteurization that is fixed fixed quantity and a fixed temperature of on which we can put the substance, and after that suddenly or immediate cooling we will follow, so by which due to the osmotic differentiation of the organism will kill. Okay, so for this we are using the holding method and flash method. Okay, so by the pasteurization technique we can sterilize the milk products and beer something also, some other product also. Okay, so by which this method some resistant kind of organism cannot destroy, like Mycobacterium tuberculosis and some species of the Clostridia also. Okay, and aseptic processing, that is relatively new commercial development in the food industry, that is most acceptable. Then in this the food item is commercially sterilized and packed into the previously sterilized container under the aseptic condition. Okay, and this process when this advantage that we are the container which we are using that is good other than cane, and that is a that provides a significant economic and use of different things. Okay, some time for the prevention of the spoilage we are using low temperature also, and sometime we will use dehydration procedure in which we will remove their moisture, so the microorganisms are unable to grow in that medium. Sometime we will destroy their osmotic pressure, and after disturbance of the osmotic pressure organism will cannot retain or cannot able to survive in that medium, so organism will die. In addition to the for the preservation we are using sometimes some chemicals for preservation, and like pickle we are using some substances, in some sauces we are using sugar and this also salt also, and sometime different radiations are also used, like ultraviolet radiations are mainly important. Okay, so these are the method by which we can prevent this. You can see that is of use of low temperature, so use of different type of chemicals in the bread and different type of things. We can use the different type of radiation like ultraviolet light, that is also in the bakeries, that is good one. Okay, as we discussed previously, the spoilage is also very important. So we have to have an idea about what kind of some things are there, what kind of spoilage, that we have discussed in previously in same lecture, and after learning about the spoilage we have just given you an idea about that how we can prevent all these things. Okay, so in the today's lecture we have just have an idea. I tried to give you some brief introduction about what kind of contamination, what kind of preservation, and what kind of spoilage the food is generally facing, and what kind of organisms is responsible for all these things. So I hope you are able to understand all the facts. So thank you very much for being with me. Today I am taking a lecture on production strains, isolation and screening technique of different microorganisms. So first we will discuss about the screening. In this topic we will also study the different type of fermentation media and fermentation technology by which we can get the different type of industrial used strain, what are the technique which we are doing for the bulk production of different products which is useful in our society. So first I want to just give the idea about what is screening. So we can say that screening is a procedure of isolation, detection and separation of microorganisms of our interest and from a mixed population by using high selectively procedure, that is called the screening. Okay, so first is isolation, is desired organism, for the isolation of desired organism. So we have to require some things in which the screening is very important procedure. So according to this, the procedure of isolation, what is it? Detection and separation of the microorganisms of our interest from the mixed population by using high selective procedure, that is called screening. That means there is a group of microorganism, but according to our need we have to first isolate and select them, these organisms, and further after separation we should have an idea about that what organism is is responsible and what organism we can use it for the different type of procedures. So first we have to take their screening, then after their isolation, then further is cultivation. So kindly see this flow diagram. Microorganisms, source where it can be the food, milk, cheese, environment, air, water, soil, maybe compost and other sources, and we will obtain the sample, or we will obtain the started organism from all these sources, and after that we will screen the organism according to the use. Then the screening takes place, the primary and secondary screening technique. So the main important things to be considered while the screening is what kind of source or what kind of things from which we are isolating the organisms. So choice of source, that is of sample from the screening, that may be from the soil, water, air, milk and composite. Next, on which the organism have to retain and use for the purpose, that is nutrients, nutrient growth. It should be supplied for the growth of desired microorganisms. And next is choice of detection, that is of proper isolation and detection of desired microorganism, that is very much important. Now, what are the type of screening? Screening is categorized into the two groups. First is called the primary screening, second one is of secondary screening. So the very much important is the primary screening, in which organic acid producing microorganisms by using of dye, and antibiotics production microorganism by the uses of Crawford plate technique, and extracellular metabolic producing microorganism by the angiographic technique, and enrichment culture technique by Griffith media. So in the first we have isolated these four categories, like what we have to make from the substrate and what kind of organism is required. First in this procedure we can first make the flow diagram and use for the bulk production. So the primary screening, that is a process for detection and isolation of microorganisms of other interest, and next is determines which microorganisms are able to produce the component and does not provide much idea about the production or yield or potential of the microorganisms. Next is separate out only the few microorganisms only. Few have a commercial values, while other organisms we have to discard, and mainly useless microorganism for that is not useful for our bulk production. So mainly the screening procedure is the procedure in which we will isolate our desired microorganism for their commercial use and for our purpose. Okay. Primary screening of organic acid producing microorganisms. First category is the pH, that is indicating dyes that may be useful for the detecting the microorganisms. and they have a capacity to produce the organic acid. Next is the dyes that undergoes the color changes according to its pH. Dyes such as neutral red, bromothermal blue that we are added to the poorly buffered nutrient agar media. Colonies are subculture after that to make the stock culture. Further testing is needed since inorganic acid base are also metabolite produce microgrowth. Now, incorporation of the calcium carbonate in the medium, that is also used to screen the organic acid producing microorganism, that is on the basis of formation of the clear zone of the dissolving CaCO3 around the colonies. You can see that in this diagram, the primary screening of the organic acid production. You can see that nutrient agar with calcium carbonate. After that, calcium carbonate will change into the CaCO3, that forming the fog around the colonies. Next is primary screening of antibiotic producing microorganisms, that is crowded plate technique, and that is used for the screening of antibiotic producing microorganisms. That means when the group of the microorganisms is present in the culture, so we will only isolate and screen out the some organism that is useful for the antibiotic production, other organism we will discard it. That does not information about the sensitivity of the antibiotic towards the microorganisms. Next, dilutions are made and then pouring and spreading of soil sample that gives 300 to 400 or more colonies per plate. Okay, the colonies that are showing as antibiotic activities are indicated by the zone of inhibition around the colonies. If the organism is susceptible to the antibiotics, the clear zone is appeared, and if the organisms are resistant, there may be the opaque kind of solution, opaque kind of we can say the structure, or we can say the formation around the colony. We can see that such colonies are subcultured. After that, they will purified by the streaking before making the stock culture. You can see that purified culture are then tested to final microbiological inhibition. You can see that spread roll during the dilution on the plate of the selective medium. The sterile glass spreader then incubate the colonies of Streptomyces species. Next is overlays with an indicator organisms, incubate. Okay. Next is non-producing organism, zone of growth inhibition, that is producing the organisms. You can see that. See that. This type of growth is appeared. Third is primary screening extracellular metabolite producing microorganism. Okay. Oxidography technique that is employed for the detection of the microorganisms that is able to produce the growth factors, add vitamins, amino acids, and extracellular substances. In this technique, we have two major steps. First is preparation of the first plate, and second is preparation of second plate. In the preparation of the first plate, what we will do, a filter paper strip is put across the bottom of the petri dish. The nutrient agar is prepared and poured on the paper dish and allowed to solidify. Then solid sample is diluted and proper dilutions are inoculated. And in the preparation of the second plate, the minimal media lacking the growth factors is prepared and seeded with the test organisms. The seeded medium is poured into the fresh petri plates and the plate is allowed to set. Now, the agar in the first plate is then lifted and placed on the second plate without inverting. The growth factor that is produced on the agar can diffuse into the lower layer that have an organisms. That means that have contains the test organisms. And third one is called zone of stimulated growth, that is made up of test organism. and that is of around the colonies, that indicates the organism produce the growth factor, which is extracellularly. The fourth one is called the enrichment culture technique, that is designed by the Wiesenkind to isolate and desired microorganisms from the heterogeneous microbial population. That means the group of the microorganisms is present, so by which this special type of technique, that is Bazering technique, is there, that is used for the isolation of the organism. That is consist of following step, that is made up of following step. First is nutrient broth is inoculated with microbial source, that is material, and incubated. Second is a small portion of the all inoculum is a plate into the solid medium, and well isolated colonies are obtained. Next is a suspected colonies from the plates are subcultured on the fresh media and subjected for the further testing. You can see that enrichment culture, in which first is, that is medium containing the selective medium that is of nutrient agar, and we have incorporated the organisms, and then second one is a sample that contain the wide variety of organisms, that is include the organisms that we want to spread. And third is organism of interest, they will multiply, and finally we will pour into the petri dish, so that organism is settled on the petri dish and form a pure culture. And this pure culture that is made, that can be only be possible by the screening method. Now secondary screening, that is a systemic screening program that is also used for the isolation of the industrial important useful microorganisms. So some important points that is associated with the secondary screenings are mainly that is useful in the sorting of microorganisms that have a real commercial value, and next is a microorganism that have a poor fermentation process, and third one is that also provide the information whether the product formed by the microorganism is new or not, and that is accomplished by the paper thin layer and chromatographic technique. Okay, it should show whether the product possesses the physical properties, that is of UV light absorption or the fluorescence or the chemical properties, and that can be employed for the detection of the different type of compound during the paper chromatography. That is also conducted on the agar plates, that is in flask or in the small fermentation containing liquid media, and by this technique we can have an idea about that whether the economic position of the fermentation and the process that involved is used to the newly discovered culture. It helps in the providing the information that is regarding the product yield potential to different organism. Okay, next is determination of primary condition for the growth of accumulation of the product associated with a particular culture. Next is a chemical, physical and biological property of the product. We can also see determined by the secondary screening method, and detect gross genetic instability in the microbiculture. We can also study about that, and this type of information is very important since the microorganisms is go the under mutation and different type of their strain is changed due to the some condition, atmospheric condition, and they absolutely lose their original character. Okay, and maximum accumulation of the fermentation product, and that shows that chemical stability of the fermentation product, and it can be quantitative or qualitative in its approaches. Before we are proceeding to further, we have to just think about that what is a fermentation, and what are the media which is used for the fermentation, and what are the major fermentation product, and what is the design of fermenter, and what is a component of fermenter. So in short we will study before we will proceed. Okay, so what is a fermentation? Fermentation is the process of growing microorganisms in the nutrient media, and by the maintaining the physiological and chemical conditions, that is their role is to converting the some substances into the desired end product. Okay, fermentation technology, that is used in use of organism to produce the food, pharmaceuticals and alcoholic beverages on the large scale industries. And the basic principle that involves the industrial fermentation technology is that organisms are grown under the suitable condition by providing the raw material, meeting all necessary requirements such as carbon, nitrogen, salts, trace elements, vitamins, and the end product that is formed by the result of their metabolism, and that is accumulated at their lifespan, that is released into the media, which are extracted for the use of human being, and then that have a high commercial value also. The major fermentation products that is useful in our industries are mainly the ethanol, lactic acid, alpha amylase, protease, lipase, some antibiotics like penicillin, streptomycin, chloramphenicol, some vitamins like riboflavin, vitamin B12, and the organism which forms these substances, we can include that first is a ethanol and lactic acid products. The organism which is used is a Saccharomyces cerevisiae and Lactobacillus vulgaris. Okay, for the enzymes, Bacillus subtilis and Bacillus species. For the antibiotics, for the penicillin, streptomycin and chloramphenicol. So for the penicillin, the organism that is used is Penicillium chrysogenum. For the streptomycin, Streptomyces granis. For the chloramphenicol, Streptomyces venezuelae. And for the vitamin riboflavin and vitamin B12, the commercially substance that is Asiaga grapsi and Pseudomonas dendrophyllus can be used. So the design of fermenter which is used for the fermentation technology is, you should have an idea about that. So what is this? The design of fermenter is the fermentation process that requires the fermenter for the successful production. So the fermenter is a large vessel that is containing considerable quantities of nutrient media by maintaining the favorable conditions, and its design and nature of the fermenter varies upon which is a requirement. Okay, so we in the fermentation container, different type of facilities are there for the process of substances, like contamination free environment. They will provide the organism contamination free environment and specific temperature maintenance and maintenance of agitation and aeration. And next is a pH control, also monitoring the dissolving oxygen, that we can say the DO. The port for the nutrient and reagent feeding, that is of antifoam agents, alkali agents and acidic agents, and ports of inoculation and sampling, and provides the all aseptic condition at time of sample withdrawn and addition to inoculum, and complete the removal of the broth from the tank, and should be easy to clean. It should be designed in such a way that it consumes less power, having a less evaporation, that can be used for the long periods of operations. And what is the component of fermenter? Mainly the component is basic component, that is a drive motor, heaters, pumps, vesicles and other accessories like peripheral equipment, reagent bottles and instrumentation and sensors. And we will also we have an also monitoring and controlling parts of the fermenter. The various type of fermenter which we are using for the industries are depends on their requirement, their size, their shape. Mainly the fermenter are can be the range of 50 liter to 1000 liter. And recently and briefly we can say the fermentants are categorized into the two type, that is surface fermentation, and second one is called the submerged fermentation. The surface fermenter is a tray fermenter, and second is a packed bag fermenter, and submerged fermenter is a simple and forward fermentation and airlift fermentation also. So if we are talking about the types of fermenter, so we can say that it can be the surface fermenter, tray fermenter, and in the first, that is called the surface fermenter, that is used for the production of citric acid from Aspergillus niger and nicotinic acid from the Aspergillus dorus. And about the tray fermenters, that is a very simplest and widely used fermenter, and basic part is made up of wooden, metal and plastic tray, and generally used for the inoculation and occasional mixture and some other things also. Okay. And tray fermenter, that is solid versus liquid medium, we can use it in this. And the liquid medium, cells are allowed to float easily into the process, and for the solid medium that is used, microorganisms are allowed grow on the moist solid substances. Solid state fermentation, that is SSF, and SSF defined as a growth of microorganisms, and advantages that produce the higher yield and possibility of contamination by the bacteria, and disadvantage is that is that causes a problem in the monitoring of the process and parameters, and some automation is also required, that but difficult to maintain. And packed bed fermenters, so they are the type of surface cellular bioreactor, and solid, mainly the nutrient broth flows continuously over the immobilized biocatalyst. And sometimes submerged fermenters we can use is, they are of microorganisms that are dispersed in the liquid nutrient medium. Okay. And next type of fermenter is, we can say that mechanical stir fermenter. These are equipped with a mechanical agitator and maintained homogeneously and rapid uh, dispensation and mixing of the material. That is, example includes the stir tanks fermenter, batches and continuous operated. Next is STF, that is stir tank fermenter. Next is continuous stir tank fermenter. Then continuous stir tank fermenter in fresh medium, that is added continuously in the fermenter vessel. On the other end, the medium is withdrawn from the recovery of fermented products. Okay. Next is continuous stir tank fermenter, that is CSTF, that is of different type of container fermentation are the single stage recycle continuous fermenter. The single stage is single fermenter that is inoculated and capped in the continuous operation by the balancing the input and output culture. CSTF and STF, they have both their advantage and disadvantage also, and sometimes the bubble column fermenters we are also using. In this bubble column bioreactor, or the air gas that is introduced at the basis of the column, and some pipes are also there. The flow rate of the air and gases are also there within this type of fermenter. In this, airlift and airlift fermenters have a different type of requirements and different type of categories also. Okay. In all these process, the total 4 phases are take place by the fermentator organism. Like, they have an lag phase, in which the immediate after inoculation there is no increase in the number of microbial cell sometime, and the period is called as lag phase. In this phase, the organism adjusts to the new environment in which they have inoculated. Second one is called as acceleration period, that is a period in which the cells just start increasing the number, is known as acceleration phase. And third, we can say the log phase. This is a time period which a cell numbers stratified their increase. And fourth one is declaration phase, that is duration which the steady growth declines. And next is a stationary phase where there is no change in the microbial cell number in the stationary phase, and the phase is attended due to the depletion of the carbon source. And finally, the death phase, the period in which the cell number decreases steadily in the death phase. This is due to the death of the cell because the essential of the metabolic activities, that is depends upon the product required for the different phases. Okay, so that is a total growth of these organisms. Okay, according to this, the fab batch fermentation and batches of fermentation that is concluded, and the time is also that on that is started, that sets in the all these type of fermenter. After the process, we can finally the process in which the death are take place by the death are take place of microorganisms. So we will conclude that fermentation process is over. So in the different type of fermentation method, some media are very much important. These media plays very much important role on different type, because media contains a different type of nutrient substance like carbon and nitrogen source, energy source, and some they will produce into the biomass production and carbon dioxide and H2O. Okay, and some nitrogen source in the form of yeast extract, carbon source in form of different food substances, some minerals, some chelators, and some growth vitamin and growth factors also, and water. Okay, some type of media that is used for the fermentation is defined versus undefined media, common, broadly and defined culture media, nutrient media, minimal media, selective media, differential media, transport media, and enrichment media. Mainly mannitol salt agar is used, sometime McConkey agar is used, and sometime potato dextrose agar is also used for the checking of bulk material. Okay, and after checking, and we will conclude that which type of media is required and what type of resources of food is required. Okay, so different type of production of the antibiotic sensitivity and some other substances, and chemical and physical and biological properties of products, that is also determined by the secondary screening. And moreover, it reveals whether the product procedure in the culture broth occurs in the more than one chemical form. And some gross genetic microbial culture that is also used for the production of different type of antibiotics. Mainly the media which is used for the growing of different organisms is nutrient agar, and after growing on the nutrient agar, we will use some selective media. These selective media on which we will obtain the pure culture colonies, and for the growing of some anthrax organism which is responsible for the production of fermentation. So we will also incorporate the McConkey media that have a sodium taurocholate and glycolate for that. And for the yeast like Saccharomyces and some other yeast fungi, and for the making of antibiotics, we are using for the growth of fungi potato dextrose agar. So the potato dextrose agar is mainly the general purpose medium, that is for the yeast and molds growth, and that is made up of potato infusion, dextrose agar, and distilled water. And when maximum, what is a medium optimization? after inoculation of organisms, and we will acquire some growth and some considering biomass growth phase, that is in isolation. It must be recognized that efficiently grown biomass produced by the optimized height producing growth phase and not necessarily based study to ultimate products such as synthesis and desired products. So the clinical design is also very much important after growing of organism. We can also categorize these product according to their requirement. And for the after the isolation of the organism, we will blanket Barman design statistical optimized techniques we are using for the further, that is used for the more than 5 variable in this, and that is very useful for the screening of the most important viable organisms. Okay, number of the experiments in which we can put on the formula, and that was concluded for the n to 1 variable, which is n is a multiple of 4, like 8, 12, 16, 20, like that. And formula that is given by some scientists, that is a series of experimental design that is known as balance this, and variable that is not have been influenced, and dummy variables are required to estimate the error. Okay, so more can be included if the real variables are less. So commercial process is very much complicated, in which first technique is screening and different type of media using different type of fermenter, and screening and isolation and obtaining of the pure culture. Then finally we have to make some statistical analysis also, by which we can obtain certain substances. So example of secondary screening is a antibiotic producing Streptomyces species. Streptomyces is a species that is also isolated by the streaking, by the narrow band of the nutrient agar plate, and then test organisms are streaked from the edge of plates without touching the Streptomyces isolate. Okay. At the end of incubation, growth inhibitory zones of each organism will be measured, and we will finalize that antibiotics, the made product that is called the antibiotic is good enough, that is useful. Okay, so this is about the today's lecture. Hope you can understand you have an ability to understand, and you are able to understand what today I have taught. Thank you very much for being with me. Today I am taking a lecture on the fermentation of different products. Okay, first I have to understand, and we have to understand what is fermentation and what is the technique of fermentation and how they are useful in our industries. So fermentation has been majorly way for the preserving the food and microbial growth either of the in the natural environment, or sometime it may be inoculated into the food medium artificially as a inoculum of some fermentable substances. Okay, so after fermentation the chemical and physiological texture of the product get changed, and that is useful for the edible items. So we can say that fermentation process also be used to create new and pleasing food and flavorous and good odor foods. Okay, mainly the fermentation used in the food microbiology for the preparation of lactic acid, propionic acid, ethanol, and some time for the different edible items. for like milk, like dosa, idli, and some other things. Okay, so uh, first we will discuss that what is fermentation. So the fermentation process is the breakdown of the starch so that they can make more metabolized by the body. In this PowerPoint, one slide is showing about how the idli is ferment. Like in the idli, when we prepare in the bulk, so what we do, we will take some black gram dal, and we will do the washing and soak it for the 5 to 10 hours. Then finally it will grind it, and in the second bowl we can take the polished rice, wash it, and soak it for the 5 to 10 hours. Then we all the things we can grind it and mix together. Okay, and add some yeast, or sometime we can use the salt also, and incubate for the overnight in the warm place. If the warm place is there, there is no need of the yeast powder. If they are in the non-warm place, that means if the temperature is cold, almost about 10 to 10 or below 10, so we have to use the yeast. Then finally we will pour the batter and make the idlis. So normally the rice and some background dals that will change into the fluffy batters, that is called the fermentation. So different type of fermentation food we are enjoying nowadays as a commercial use also. So nowadays some fermented milks are also there, and they have containing the different milk products, that means of lactic acid fermentation, and sometime the yeast lactic fermentation, and sometime the mold lactic fermentation. Okay, so the lactic fermentation, that is of mesophilic and thermophiles, and they are therapeutic also. So the in case of milk, buttermilk is a mesothermophilic and mesophilic, and colored buttermilk, and some other flavored things. And next is thermophilic, that is of yogurt, and some labin, and therapeutic, that is biodegrade, biofingers, acidic milk, yogurt, Yakult. Nowadays one milk is very much famous, that is called the Yakult. That is also a therapeutic use, because we are using for the treating of lactobacilli in the intestine. And yeast lactic acid fermentation, that is of yeast milk, and mold lactic fermentation, that is villi. Okay, so that is of different type of fermentating products, and which we are using for the edible or as well as for the industrial purpose also. So in the dairy industries, it is very important, and sometimes we are using some microorganisms as a starter culture, and then can be related to process, and we will obtain the desired fermentation product and good quantity product. Sometimes Streptococcus thermophilus, Lactobacilli, and Lactobacillus bulgaris is also useful. They also useful for the preparing the yogurt from the milk. A lot variety, several thousand varieties of cheese that are manufactured nowadays is a good food products, that is also made up of by the different kind of microorganisms. Okay, so we have discussed some microorganism, but other fermented food, like we can also mention here is, we have to include that in this is pickles, and olives, and certain type of sauces. Okay, so lactic acid bacteria are chiefly responsible for the desirable type of fermentation that is required for the fermentation. Okay, the microorganism that produce the changes may be natural flora on the material to be fermented, or maybe something else that is added as a starter culture. Most commercial sour, sweet, and mustard and Mixed pickles are made up of from fermented salt soaked pickle. And other major type of pickled cucumber is fermented dill pickle. Okay, so that is a that we can use it as a commercial fermentation process. And different type of other food substances we can also just a minute mentioned here. So the microorganism that is of as a single cell protein we can discuss here. Just a minute. Different type of procedures which we are using as an fermentation. So first we are discussing about the pickles. So the pickling process is preserving the fruit or anaerobic fermentation. We can use it by the brine and vinegars. Sometimes we are according to our taste and according to requirement of the industries we can pour it the salty and sour taste also. And fermentation cucumber is very similar to for the making of the square and fermentation where only the brine and salt solution is used instead of the dry salts. Okay, so large tank we are using in which we will use the 15 to 20% of sodium chloride. Okay, and this brine is used. Brine is what brine is a salty solution in which we can make the pickles and we can preserve some other things also. Now another distinguished character of the pH that should be of less than 4.6, which is sufficient to kill the most of bacteria. And pickling can preserve the uh, perishable foods for a month. Antimicrobial herbs and spices such as mustard seeds, garlic, cinnamon, and cloves. Okay, the food contains sufficient moisture. Pickling brine may be produced simple by adding the salts. As I told you previously that natural fermentation occurs at the room temperature, that is due to the lactic acid bacteria. And for the making of pickles that is more than enough. And by the other pickles we have to use the vinegar also. And sometime for the canning process for the fermentation and for the making them style we have to use the vinegar. Okay, when both salt concentration and temperature are low, so leucocytic substances like mix of acid, alcohol and aroma compounds we are using. Sometimes the high temperature we are using, then we will use the lactobacillus and lactic acid. So the mainly pickle start with an leuconos and lactobacillus with higher acidity. Okay. Now Sauerkraut are the German term that is for the cabbage. And you know that the product of the natural lactic acid fermentation of the fresh cabbage in the presence of a small amount of the salt, if that is included the lactic acid bacteria, so we can made it. That is sauerkraut is rich in the vitamin C. Okay, and mainly the nutritional value is about energy and carbohydrate almost is about 4.3 gram and sugar is about 1.8 grams and dietary fibers are also there. And you can see there then some fat molecule, fat substances, water, vitamin D, vitamin B, C and iron, sodium components are also uh, here for the good health. Okay, long shelf life and different type of flavor and taste, that is very much important, that is used to the different type of microorganism which we are using in this for the preparations. Next item is bread. Bread is also commercially now that is very good in use. So the bread are generally made up of yeast. First we make the dough. After that in dough we will inoculate some yeast component, water, salts. So the yeast ferment and some sugar compound, some component also we will add. So these yeast will utilize the sugar and form some gases and alcohol, and so that gases make them fluffy, and some alcohol that will change the taste of dough into the good flavor. So that is called the, and after making this, we will commercially use and use for the different type of recipes also. Now you can see that how we can make the bread, that is mixing of ingredients and raising, fermentation and kneading, then molding into the loaf shape, then raising, then baking, then cooling. After cooling, we will cut into the slice. And now the making of vinegars, as you know that vinegar is also the liquid consistency, that is of acetic acid and water, and acetic acid is produced by the fermentation of ethanol by acetic acid bacteria. Now that is also very much used for the cooking purpose, and used in the as an in bulk amount in the food industries also. Okay. And commercially vinegar that is produced either by the fast or the slow fermentation process. In general, slow method are used within a traditional vinegar, and fast fermentation we can make it by the commercial methods. The longer fermentation period allows the accumulation of the non-toxic slime composed of acetic acid bacteria. For the fast method, adding of the mother of the vinegar, that is bacterium cultures, to be the some sources of the liquid before adding air. Okay. So in the fast production process, vinegar may be produced in the period ranging from the 20 hours, 24 hours to the 3 days. Now, how we can make the vinegar? Like mango, washing and peel off, and mechanic pressure, mango juice, then pasteurization of the concentration of the total sugar and contents of the juice of the 20 weeks, and alcoholic fermentation, acidic fermentation, mango vinegar. And in between that, we can also add the yeast and acidic bacteria, and determination of the yeast and determination of the bacteria biomass. Vinegar is commonly used in the food preparations like making of the pickles and some other salad dressing. Somewhere it is also used in a therapeutic use, like making of the some substances that causes the blood glucose control, and used for the diabetic management also. Okay. The making of idli, we have initially we have studied about the idli, that is a main traditional disease of the South India, and we in this the black lentils is used for the fermentation process, and some other component that are made up of by fermented substances. So we can count is there is an account cultured buttermilk, cultured sour cream, and Bulgarian milk, acidophilus milk, and yogurt, kefir, kumis, and some sauerkraut, and pickles, green olives, some sauces. These are the fermented products, and we will study about some detail what are the substance on which what kind of bacteria is causing the effect. After that, the product is formed. Okay, so in the culture buttermilk, the mixture of lactic acid streptococci is there. So streptococcus lactis and S. cremonis, that produces the aroma of the bacteria. Okay, the function of the lactic acid, that is streptococci, is to produce the lactic acid, and that gives the sour taste and to curdles the milk. The function of the leuconostocs is to provide the volatile and natural products that impart a characteristic desirable odor, and the starter culture must have starter culture containing the a lot of growth of bacteria, and the incubation, that is incubation temperature is 21 degrees centigrade. And about the cultured sold milk uh, or the cultured buttermilk, mainly the organism which are responsible is the streptococci and leuconostocs. No strictly fermented milk is there, but manufacturer resembles that culture buttermilk. The cream is inoculated and incubated until the desired acidity develops. So, after the inoculation of the organism, some flavor and aroma compounds are also generated by the using of the starter culture. And next is a Bulgarian milk. So, in the Bulgarian milk, the causative, the inoculating organisms are is which is used for this preparation is a Lactobacillus bulgaris, and for the milk incubation period is 37 degrees centigrade. Otherwise, the simple culture buttermilk, that products from the different commercial buttermilks, and heavily acidic atmosphere is made, and in this Bulgarian milk there is an aroma is not present, and in acidophilic milk Lactobacillus acidophilus organism is responsible, and milk for the propagation of the L. acidophilus, and the buttermilk is fermented and sterilized. An organism is first they overgrown, then other contaminating organisms are also be there, and the incubation period is about the 37 degrees centigrade, and acidities allow them to develop them into the media, and acidity is developed up to the 0.6 to 0.7% after growing of the organisms. Okay, next product is we can include is yogurt. For the making of the yogurts, Streptococcus thermophilus and L. bulgaris is used. That is made from the milk in which the solid and concentrated some milk products are there, and some water is we will added, and some skim milk, and kefir, that is main principal microorganism that is responsible for the fermentation is a lactic acid, Streptococcus lactis, and some Lactobacillus bulgaris, lactose fermenting yeast. Okay, and generally the mixed lactic acid and alcoholic fermentation is going on in this product, and acidity is maintaining up to the 0.6 to 1%. Okay, the organism that forms the organism is inoculated as a granules, and finally the kefir is formed, and that is made from the cow, goat, and sheep milk. Okay, next product is a kumis, the similar to found in the kefir granules, that is the mixture of the acidic alcohol fermentation products from the mare's milk in some parts of the Russia. Kumis is also made up of different type of milk. According to the milk, the organism we are using, and they will ferment the organism and they will produce the taste and odor for that. For the some green olives, the fermented fruit, the green olives, the starting products is olives, and the organism is Lactobacillus mainly group, mainly brevis and platinum, and some mantroids are also used for the fermentation of these green olives, and some sauces like starting product is beef and pork, and substances which is used for the sauces are uh, pediococcus and micrococcus species that is used for the making of different products. Here we have discussed about the different food products and the responsible organism and some substrate on which the organism um, work, and they will produce the different order and product for the eating and as in a food industries. Okay, so how they will form the single cell protein, that is of the dried cell of the microorganisms, and some bacteria, yeast and algae, that is produces the massive quantities, and they attracts the sucrose of the food for the animals and for the human also. The microorganisms can be cultivated or on industrial waste or on the byproducts also as a nutrient and large cell crop and rich in the protein, that is called the single cell protein. On bacterial cell grow on the hydrocarbon waste from the petroleum industries. They are source of protein in the different type of crop. Okay, and the yeast cell crops harvested from the vats using and producing different type of alcoholic beverages that have been used as a food supplement and for the generation of the different type of food. Got it? Then single cell protein as a food substitute or the substance, that is also very good, and we are we can make it by the using of the different type of microorganisms. And microorganisms grow very rapidly and produce a high yield, and it has been calculated that one can obtain the again 1 lb of the protein in the 1 day's growth is about the 1000 lb stairs. You can see that in the very short duration a lot of organism we can generate and used as a different type of starters and some as a food content also. Now, like some spirulina, that is rich source of the protein about 60 to 70%, that includes the vitamins, amino acids, minerals and some curd like substances, that is also be there. And some multiple spirulina that offers several advantages over the single cell, and some algae that is also good for the eating purposes, and it's sorry, some microorganism that is also useful for the preparation of cheese production. Okay, so cheese is one of the oldest human food, and it has been developed approximately 8000 years ago. Okay, and different and distinct varieties of cheese we can produce by the using of different species of the microorganism. Almost in India 20 general types we can use of the cheeses. Okay, so how many major type of cheese and what type of microorganism we will discuss here. Like sauce, soft and unripened, that is called the cottage and cream and mozzarella cheese, that is made up of lactobacillus lactis and some other species like L. cremonis and L. vulgaris. Okay, and production is Leucosteria cremonis. And second type of cheese, that is of soft and ripened, that is called the brie. And brie second name is also we can mention here as an France, and Camini battle, that is also from the name of France. The main organism that is lactobacillus species, mainly involve the lactocremonis and L. lactis. For the making of semi-soft cheese, the different variants which is available are blue and Roquefort cheese, that is also mainly used in the France, and brick and monster, that is used in the main tagging of this type of cheeses about the United States, and Limburgers, that is Belgium type of cheese. And all type of these cheese are made up of lactobacillus organism, Lactobacillus cremonis, Lactobacillus lactis, and Lactobacillus cremonis is majorly used. And later stage of the production is a penicillinum and a brevibacterium. Next type of cheese is hard and ripened cheese, that are called as Cheddar cheese, Colby cheese, that is used for the Britain preparations. And main the organism which is produce these cheese are Lactobacillus lactis and also the Cremanis, and later stage this production is taken up by the Lactobacillus casei. Okay, and Swiss cheese is primarily earlier stage production, that is of Lactobacillus lactis and Helvenis, and finally that is taken up by the Propionibacterium. Okay, very hard and ripened cheese, that is also called as Parmesan, that is of Italy cheese, and mainly the causative, the preparing agent is Lactobacillus lactis, Cremanis, Thermophilus, and further that is taken up by the Lactobacillus vulgaris. So these are the different type of cheese and related type of microorganism which causes which helps in the formation of different type of cheese. So microorganisms are also very much useful in the food industry as a manufacturing of the cheese also. Like meat and the fishes, beside the fermentation of the dairy product, variety of meat products, especially sauces, can be fermented, and country curd hams, summer sauces, salami, and cervelle, and Lebanon bolonga fish sauces, that is processed by the halophilus bacterial species like Bacillus species, Izushi, and Katsubichi, that is of Pedicococcus cerevisiae and Lactobacillus plantarum, that is most of them involved in the sauces fermentation. And Izushi is based on the fermentation of the fresh fish, rice, and vegetable by the Lactobacillus species. And Katusobish result from the fermentation of tuna by Aspergillus glansu, both meat fermentation, that is originated from basically from the Japan. And some alcoholic beverages, that is also is a product of fermentation, so that is also commercially used. So in the commercially preparation of the different type of alcohol and wines, these organisms are responsible. Okay, so variety of the plants that have an adequate carbohydrate can use for the alcoholic beverages. When carbohydrate is available in the readily fermentable form, the fermentation can start immediately. Like grapes, when we first crush in to release the juice or the must, we can allow them to ferment without further delay. Mainly the organism, first we have to sterilize by the pasteurization and by the use of sulfur dioxide, then desired microbial growth, that is useful for the making of alcoholic beverages, we will add it as a starter. Okay, in contrast, before cereals and other starchy material can be used for the substrate for the production of the alcohol. Their complex carbohydrate, they are hydrolyzed, then they are mixed with the water and incubate the process that is called the mashing. Okay, after after doing the mashing, the insoluble material that is removed from that is called the wort, and clear liquid that is containing the fermentable sugars and other simple molecules. Okay, then formation of the beer and production of the different type of wines and champagnes is used different type of microorganisms for their productions. For the wine production or the Greek onion and wine production, we will also use some grapes and some organism as a starter. Okay, like grapes with white juices, and for the making of red wine, red grapes, and we will take out their skin, then make the pulp. After the natural fixing of, then we will inoculate some bacteria and yeast, so they will produce some fermentation, and the organism which is used for the making them according to your need is mainly Saccharomyces cerevisiae and Saccharomyces ellipsoideus. After the incubation, the juice is fermented for 3 to 5 days at the temperature varying from 20 to 28 degrees centigrade. How we want to manufacture the alcoholic beverages, that is depends. Okay, like depending on the alcoholic tolerance, the yeast starts the final production may contain 10 to 18% alcohol, and um, mainly the organism is a leucostate oceum and S. plantinum, L. brevis and L. casei. These are the mainly organism that is used, and these microbes that they transform the malic acid to the lactic acid and carbon dioxide. The result, the pH is increased, and further the improvement of the flavor stability. Some organisms and some yeast substances we are also incorporated. The critical part of the wine making involves the choice of whether the product is produced. We have to produce the dry or retaining free sugar or the sweetener. So according to that, we will choose the organism. The many processing variations during the production. The wine can be distilled to make the burnt wine or the brandy. The acetobacter and gluconobacter can be allowed to oxidize the ethanol to the acetic acid, and that will change into the wine vinegar. So by the using of different type of organisms, we can make lot of substances and lot of food items that is useful as an dairy industries, as different type of alcoholic industries. So today we have discussed about the fermentation, different products, how we can make by the using of fermentation techniques. I hope you are able to understand this fermentation and different type of organism, how they work on the substrate and produce some commercially available food substance and other products. So thank you very much for being with me.